I want to highlight the importance of the structure of molecules, why we have to know the exact structure of the molecules, why it is important to us, what skills are needed to understand the molecules, in particular biomolecules. All living things are made up of four classes of compounds, biologically important ones: carbohydrates, lipids, nucleic acids, proteins, etc. When you come to biomolecules, we'll have the different varieties of classification based on the size and molecular weight. Okay, inorganic minerals, gases, waters will be there. Similarly, organic units, simple sugars, amino acids, nucleic acids will be there as micro biomolecules. There are large biomolecules are there whose size is more than 1000 daltons. Carbohydrates, monosaccharide, except monosaccharide, disaccharide, trisaccharide, polysaccharides, lipids, proteins, nucleic acid, you can have their classification. When you come to the carbohydrates, carbohydrates are primarily produced by the plant and form a very large group of naturally occurring organic compounds. Most of them have general formulas CxH2OY, and they are considered as hydrates of carbon. Chemically, one can define carbohydrates as optically active polyhydroxyaldehydes or ketones are the compounds which produces such units on hydrolysis. They can be considered as carbohydrates. There further, you can have classification of these carbohydrates: monosaccharide, disaccharides, oligosaccharide, polysaccharides, etc. The simple unit of any of these biomolecules, you can say that they are called as monomers. Monomers are the simple units which are present in all these type of biomolecules, large biomolecules. Okay. These carbohydrates which are sweet in taste are also called, they are the classification. You can have sugars, non-sugars, and reducing sugars, non-reducing sugar, etc. We can have different types of carbohydrates. As I told, when you come to the classification of carbohydrates, we are having classification as monosaccharides. These carbohydrates cannot be hydrolyzed further. They are the simpler units of any sugar molecule. About 20 different types of monosaccharides are known to occur in nature. Some common examples I have taken here. You just see the structure of these molecules: glucose, fructose, galactose, deoxyribose, ribose. These are important sugar molecules. When you come to the classification of these carbohydrates, monosaccharide can be further classified as trioses based on the number of carbon atom: trioses, tetroses, pentose, hexose. Like that, you can have the classification. Another classification is based on the functional group. You can have either an aldehyde group or a keto group in the molecule. If a sugar is having an aldehyde group, we call them as aldohexoses. OSE will be there at the end of the sugar. If it is a ketone, it is called as ketohexoses. If it is having 5 carbon atom, this is having 6 carbon, so it is called as hexose. And sorry, this is having 5, so therefore it is called as pentose. This sugar is having 6, therefore it is called as aldohexose. This is ketose, pentose, ketopentose. As we know that if a molecule contains 2 units on hydrolysis, if a sugar gives 2 molecules on hydrolysis, then we say that they are called as disaccharides. So in this classification, oligosaccharides generally refers to group of carbohydrates which on hydrolysis gives 2 to 10 sugar units. Okay. When you come to disaccharide, it contains 2 sugar units. A molecule which is having 2 sugar units on hydrolysis, then we say that it's a disaccharide. Here we have taken an example of maltose. Just look at the linkage. This is one glucose unit. The another one is D-glucose. Both of them are D-glucose units. These D-glucose units are linked to one another through 1,4-glycosidic linkage. This is called a linkage, is called as glycosidic linkage. This is 1 and 4. One of the sugar is linked to the 4 of the another carbon atom. Therefore, it is called as 1,4-glycosidic linkage. Because of this linkage, this is the free anomeric oxygen atom is open, therefore it can undergo, it can show reducing property. This is a reducing sugar. Okay. The properties of these sugar molecules, they depends on the nature of the bonding, their size, their structure, okay. Physico-chemical property, they depend on the structure of the molecule. That's what I want to emphasize. Here, we'll consider lactose. This is beta lactose. This is made up of two units. One is galactose, the other one is glucose. The linkage is 1,4-glycosidic linkage. Here also, again, this oxygen atom is free. It can open up and it can behave as a reducing sugar. There is sucrose. We are having a wonderful molecule. It is sucrose. Here, anomeric carbon atom is free. This anomeric carbon atom is involved in the glycosidic linkage. One sugar is a 6-membered glucose unit, alpha D glucose, and the another sugar unit is a sucrose. Again, the difference is there. This is sweet in taste. The properties, as I told, many of the properties, they depend on solubility, okay, their physical properties, chemical properties, reactivity, their affinity towards polar and non-polar molecules. Everything they depends on the structure of the molecules. Okay, here we'll look at the sugars which are present in the blood. Okay, ABO, blood types, refers to carbohydrates and the red blood cell. See, N-acetyl glucosamine is common in all this one, and this is a galactose. The end is fructose in all the blood groups. Okay, whether it's A group or O group or B group. Here, in case of O, galactose is present. In this A group, galactose, it is linked to N-acetyl galactosamine. These are two galactose units are there. Based on the nature of the linkage of this one, the nature of your blood or the type of the blood also changes, and the same phenomenon is used while identifying the blood types. Okay, now we'll move on to polysaccharides. So as the definition, we know if more number of sugar units are linked to one another, then we call it as polysaccharides. These on hydrolysis, they will give you monosaccharides, etc. Cellulose. Just look at the linkage. Here, 1,4-glycosidic linkage continues throughout in a straight chain manner. This is cellulose, starch, glycogen. Okay. Here we are having different types of linkages. Based on the linkage, they will show the different properties. The main constituent, cellulose, is the main constituent of the supporting tissues of plant. Vegetable food contains cellulose. We cannot digest cellulose as the cellulase enzyme is not there in our system, but this cellulose material which we take in the diet helps the food residues which are present in the stomach will come out easily. Okay, this is storage polysaccharide. Starch is an storage polysaccharide present in the plants. Glycogen is animal polysaccharide. Okay, animal starch you can call. Okay, this is the reserve carbohydrate which is present in the liver and muscles. Okay, that is the thing. Next, we see the structure of this one. Here, again, 1,4-glycosidic linkage is there. This is one molecule. It is amylose and amylopectin. There are two things are there in starch. Okay, amylose as well as amylopectin. Because of this branching, okay, because of this branching, this starch is not soluble in water. Starch will not be soluble in water because of branching. The branching occurs, see, the linkage between the one branch and another is 1,6-glycosidic linkage. Okay, similarly, we'll move on to this one, cellulose. Cellulose is a linear molecule, aligned so firm, and one molecule, polymeric molecule, lined with another polymeric molecule. As a result of this, the rigidity to the structure, it gives the rigidity, whereas in the previous case, there is no rigidity. So this will be like amorphous in the nature. This is also okay, but that gives strength to the molecule, Cellulose, the structure is, it is 1,4 glycosidic linkage is goes on, and the packing is also there. Because of this packing, it will give the rigidity to the molecule. Okay, so these are the varieties I have discussed. Now straight away I will move on to the structure elucidation of glucose. Okay. See, we know that Emil Fischer is responsible for the structure elucidation of glucose, and generally you will be teaching the structure elucidation of the molecule to the students. Okay, elemental analysis, etc., molecular weight determination of the compounds. Okay, all those things will be done. Then, based on the chemical reaction, let us assume that we don't know the structure of the glucose, but glucose consumes 5 molecules of acetic anhydride, indicates that there are 5 OH groups in the glucose. It's a penta-acetyl derivative will be formed. It reacts with hydrogen cyanide, indicates that it contains an aldehyde group. Easy reaction. It reacts with hydroxylamine to form glucosamine. All these reactions indicate that glucose contains an aldehyde functional group. It undergoes easy oxidation. That also indicates that it contains an aldehyde. It's not a keto group. Nitric acid oxidation, it gives saccharic acid. Both the OH groups, 2 molecules are consumed. That means that one is aldehyde group, the other may be a primary alcohol which undergoes oxidation. Like this, we are going to draw some conclusion. So Emil Fischer has taken almost a decade, 12 years to establish the structure of glucose. That is the importance of structure is there, but they have taken large amount of time to establish the structure. This is because during that time, analysis was very, very difficult. There were no apparatus, no chemicals. They have to develop everything on their own. That was the thing. And this sugar on heating with hydroiodic acid red phosphate gives N-hexane. A normal hexane is obtained. So normal hexane is CH3, CH2, 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 CH3. That is the N-hexane is obtained. And 2-iodohexane is also there. N-hexane clearly indicates that glucose is a straight molecule containing 6 carbon atoms. That is the thing. Okay. So 6 carbon unit 1, 2, 3, 4, and it is having 5 OH group and 1 aldehyde group. Convention, we will put CHO at the top, CHO, and 5 OH groups are there. You can place it on any of the carbon atom, 5 OH groups. And as sugar is a stable molecule, that clearly says that the OH groups are not present on the same carbon atom. This is the analogy. This is the analogy we are giving. This is the skill which is needed while establishing the structure of the molecules. See, so based on that, what happened is, so OH groups are on the different carbon atom. So they have placed H, OH, 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 and the different carbon atom valency is satisfied. So you are going to get CHO, CHOH, 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 CH2OH. That is the structure of the glucose. As there are 4 asymmetric carbon atom, 2 to the power of 4, we are going to get 16 isomers for glucose. So that becomes very difficult for establishing the structure of the glucose. So for that, what they have done is they have taken glyceraldehyde. Glyceraldehyde is the simple molecule. It's a triose, right? Here, one asymmetric carbon atom is there. 2 to the power of 1 is 1 asymmetric. This OH group is on to the right, right? So if it is on to the right, it is taken as D-glyceraldehyde. Emil Fischer named it as D-glyceraldehyde, and he has added one HOH. HOH this side or this side. Okay, you are going to get 2 molecules, erythros and threos. Again, if you add HOH, okay, this is HOH, this is HOH, HOH, HOH. You are going to get 4 sugar units, that is pentoses, ribose, arabinose, xylose. Okay, the last one is xylose, lysose. Okay, so this ribose sugar on addition of HOH, that is Kiliani synthesis we carry out, okay, to introduce an HOH functional group or OH. You are going to get all these molecules. This is allose, maltose, glucose, mannose, mannose. This is glucose, iodose, galactose and talose. All together gladly made gum in the gallon tank. That you can remember to give this information. Out of this structure, one is the exact structure of the glucose. Okay, so configuration has to be established for the glucose. That is done by Emil Fischer. He has taken glyceraldehyde, Chileani synthesis is carried out, okay, to get an arabinose. Arabinose is a pentose sugar. This can give glucose and mannose. These are the two possibilities are there. Based on the knowledge of oxidation reaction, reduction reactions, degradation studies, etc., we will come to know that this is the structure of the glucose. So glucose structure is established. Open chain structure for the glucose is given. Wonderful thing. Okay. Afterwards, we found that the aldehyde group is not free. Glucose does not react with sodium bisulfate easily. Glucose does not react with ammonia easily. It will not give skips reagent test or 2,4-DNP test. That means that the aldehyde group is not free. And two isomers for sugar is formed when they are crystallized using different solvents. These things indicate that glucose exists in alpha as well as beta form, and that can be explained only on the basis of a ring structure. That means that the OH group will link to this CHO forming a ring. So this is a 6-member ring system is assigned. 6-member ring structure is written, and this is the ring structure for Haworth ring structure for glucose. Okay. So when you take alpha D glucose, the specific rotation is 111, and beta D glucose, the specific rotation is plus 19.2. When you allow the solution of alpha D glucose or beta D glucose solution to stand for some time, then the specific rotation changes, and the value will be changes to 52 degree. This is called as mutarotation. Okay, this mutarotation you can explain easily. Whatever the sugar, this will open up to form this, and ring closure will take place. While ring closure will take place, it will form either alpha glucose or beta glucose. The percentage of formation will be different. Based on that, the specific rotation changes to 50.5. So like this, we have assigned the structure for glucose, that is alpha D glucose and beta D glucose. Okay, so this much of experiments were carried out, so many analogies they have taken, so many years of experience they have put, all these put together, they were able to give the structure of the glucose, and their configuration of D glucose is also established. Okay, so almost 20 years they have taken to give the structure of this D glucose. Okay, nowadays, when you look at the technology we are having, and for that, all these reaction, to carry out all this reaction, they need kgs of the samples, 2 kg, 3 kg, 10 kgs of the samples. They might have prepared, they have taken, purified. After purification, they might have entered into this structure L solution. All this process has been established. Now we are having IR, NMR, mass spectra, and XRD studies are there. Based on this, if you are having 1 or 2 milligrams of the sample, that sample is sufficient to take the IR spectra of the compound, NMR spectra of these compounds, okay, C13 NMR spectrum, then mass spectra is there, which give you the molecular weight of the sugar molecule. This is 179.055 is there. Actually, if you calculate, it is 180.2. So you are having the fragmentations. You can see how many what are the fragments you are getting, what are the functional groups are present. All these things are easily you can identify it, and X-ray crystallography clearly shows that whatever the Haworth structure given by Haworth, okay, is correct. So these are the simple methodologies we have adopted to get the structure of the molecules. So I'll say that structure of any molecule is very important in designing so many other things that I will tell you all those things. And tests are there. Skills are needed when you are conducting the experiments also in the laboratory. The very simple test is to know that whether sugars are non-sugar. Molish test you will conduct. Take the solution. Compound in a solution form is taken, sulfuric acid is added to the sides of the test tube, then alpha naphthol is added. Okay, a purple ring is formed at the junction of the test tube that this experiment will conduct. See, if you shake the solution, you don't get the purple ring at the junction. That means the skin is very, very essential for doing many things. And ozone formation, sugars form ozone with phenyl hydrazine. When it is treated with phenyl hydrazine, they will form phenyl ozone. See, these phenyl ozone, you can prepare them, for different sugars, sugars. So needle shaped glucosone crystals. Okay, for this is for glucose. Galactosome shows this type of crystals. Powder hedgehog shaped crystals for lactose. Okay, sunflower shaped for maltose. See, by looking at the nature of the uh, ozone formed under the microscope, we can say what is that sugar. Even you can do this experiment. Okay, chromatograms, you can run the chromatogram. Okay, sugars are subjected to thin layer paper chromatogram. You will be knowing what is paper chromatography. Unknown sample can be spotted along with the this one, and knowing the RF values, you can say what is that sugar, whether it is maltose, lactose, whatever it may be. And one more very important aspect we will come across in the when it skill is needed, estimation of sugars. So this is quantitative estimation of sugar can be done using Welling solution A and B. Okay, so sugar solution is taken, Welling solution is added, it is heated. Okay, after it, mixture of Welling solution A and B. Okay, 10 ml addition of dextrose sugar is added to it, and just before the endpoint titration will be carried out. Okay, against this one. Okay, 10 ml of Welling solution is equal to 0.05 grams of glucose. Okay. So what is the amount of Welling solution is consumed can be calculated. Based on that, we can calculate what is the amount of the sugar present. All these experiments can be framed, and they can be studied very well in your laboratory also. And the reactions, you look at the reactions of this molecules. As I told, because of this structure, okay, when this is treated esterification, okay, so esterification takes place at this, and when it is reduced, okay, you are going to get reduced the sugar unit is reduced here, CH to OH, alcohols will be formed. On oxidation, that aldehyde group, okay, the aldehyde group will be converted into acid, acetic anhydride pentaacetyl derivative will be formed. When you treat it with the RX, okay, alkyl halides, okay, we are going to get alkylation of that OH group will takes place, OR will be formed like this. They undergo different types of reactions. Now we'll move on to the another set of compounds, that is lipids. Lipids are very good functional units. The lipids function as a stored energy, right? Insulation for the body and against absorption of certain vitamins. The lipids are large molecules that can be categorized as fats or oils. Lipids are composed of triglycerides. These molecules are made up of carbon, hydrogen, oxygen. Those things there, their classification, you can have lipids can be the quantitative and qualitative tests are there for lipids, solubility test, spotting test, acrolein test, saponification values are there, Lieberman Bouchard reaction for steroids. Okay, so many things you can carry out skillfully. All these things needs skill, right? All the things. And this is the lipid. This is a cell wall. Any cell wall contains a lipid. This lipid arrangement is there. Based on the structure, their arrangement is there. Okay, they can open up this membrane for the entry of carbohydrates, glycoprotein, external member. Okay, so many things they will do. There are simple lipids are there. Okay, simple lipids, they contain uh, glycerol and fatty acid. Glycerol is, this is the glycerol unit, and these are the fatty acid molecule. They will link together to form a triglycerides. So triglycerides are called as your oils or fats. So if the alcohol unit is lipids, which are long chain saturated fatty acids with long chain saturated alcohol, they are called as waxes. You are having different types of waxes. So you know the importance of this all these molecules. Okay. This complex, when you come to this complex lipid, phospholipids, They contain alcohol, fatty acid, nitrogen, containing bases and other substances. This presents in large amount in nerve tissues, brain, liver, kidney, pancreas. They contain these phospholipids. They increase fatty acid oxidation, okay, help in blood clotting, okay. These are the structural materials of cell wall, mitochondria, and microsomes. So these are responsible for the shape of the cell. When you move on to glycolipids, these mainly contain amino alcohols attached with the amide linkage and glucose and uh, carbohydrate. There will be a carbohydrate, there will be a amide linkage, okay, and these are present in medulla and nerve fibers. They are mainly present in medulla and nerve fibers. Signal transporting mechanism, all those things are involved. If this sugar, glucose or galactose, if glucose is there, its behavior different, galactose is there, behavior is different. That leads to some disorder also. Okay, liposomes. These are triglycerols, phospholipids, cholesterol, and they contain some protein also. Egg, milk, they contain phospholipids. Okay, yeah, we are having uh, 5 different types of lipoproteins are acromyl, very low density lipoprotein, low density lipoprotein, all those things are there. These are very important. They help in the transport and deliver the lipids to the tissue. So delivering of the lipids to the tissues is done by these lipoproteins, and they help in the maintenance of structural integrity of the cellular surface on the cells. Okay, and the percentage of this lipoprotein increases. Okay, in case of uh, diabetes and arthritis. Diabetes and arthritis, in case of any person is having diabetes and arthritis, the percentage of this lipoprotein content of the lipoprotein increases. One, by detecting the percentage of lipoprotein, we can say that the person is having diabetes or arthritis. They can be used as a markers. Okay, they can be used as markers for identifying the diseases. And the last one is steroids. So steroids are again, they are taken as this one, testosterone hormones are there, cortisone hormones, vitamin, cholesterol. These are all our lipids and fats. The next unit is nucleic acids. Okay, so here nucleic acid are a very important class of organic compounds and biologically important molecules because they are in the genetic material. Okay, so the monomer is here, it is nucleotides. Nucleotides are the monomers which are present in this one. So they contain phosphate group linked to a ribose sugar, deoxyribose sugar, and there will be a nitrogen base that may be adenine, guanine, cytosine, thymine, uracil, whatever it may be. These are the structure of the different nucleotides. Okay, so AMP, this is phosphate group is there, adenine monophosphate, diphosphate, triphosphate nucleotides are there, right? These are the energy units which are present in your DNA, RNA, mRNA, all those things, and responsible for various activities. Now we move on to another important class of structure of important molecules, that is proteins. Okay, proteins are chemically, proteins are polypeptide chains of amino acids. This peptide bond defines them as oligopeptide, polypeptide, etc. Oligo means few, polypeptide. Okay, oligopeptides are molecules having 2 to 10 peptide linkages there. Then polypeptide will have more than that one. If more is more, based on the molecular weight, we classify them as peptides and proteins. If the molecular weight is more than 10,000, we call them as proteins. Okay. See, all this they contain non-polar and polar molecules are there. These are the amino acids. These amino acids are the basic units which are present in the proteins, and they constitute, so this OH group, acid group of one amino acid linked to the NH2 group of another amino acid to form a poly. This one is a peptide linkage. This is called a peptide linkage, and this is a dipeptide. Okay, similarly, different number of them, they will link together to form a dipeptide and whatever. See, you just see the geometry of the molecules. So they are not planar molecules. That's what I want to say here. These molecules are not at all planar. They will have a different dimensions. They will have different orientation. You look at the hemoglobin molecule. It contains alpha chain, iron unit is there, hemine is there. So they will have a different structure. So these proteins are a very important class of compounds because the class of protein is enzymatic proteins, biological, they act as biological catalyst, urease, amylase, alcohol, so many things. Structural proteins are there, transport or carrier proteins are there, okay, hemoglobin, myoglobin, all those things, nutrient and storage proteins, okay, defense proteins is there, regulatory proteins, toxic proteins, snake venom, etc. These venoms also, they are proteins, okay, they are also used in the treatment purpose also, right? Different proteins, these are the structure of the protein molecules, okay. So simple see, amino acid linked together to one another, like this. This goes on, okay, to form a simple structure. This is called as primary structure of the amino, this one, proteins. This is the primary structure. They can undergo folding, right? Because of the hydrogen bonding, they will take this type of structure. This is beta plated structure. One unit is there, another unit is there, but there is an interaction between these two by what is that, hydrogen bonding. Okay, these are the two or secondary structure of protein. So this is tertiary structure of protein is there here. Again, different units, okay, they are having a beta plated structure and all those things, right? Okay, this is the polypeptide chain. This is quaternary structure assembled subunits. Okay, there are different subunits are there. All these, this is one unit only. This is the tertiary structure where this is continuation is there. See, one unit of protein is there. Here, number of protein units they will assemble together to form a quaternary structure. Okay, so structures are important. Identification of protein, biuret test is there, Milan's test is there. One can carry out biuret test to know that biuret test is negative, blue color, biuret positive. So we can say that they are proteins. Miller's negative test, absence of tyrosine or phenol, etc. can be identified. Okay. Once the structure of the molecule is known, what to do with the structure of the molecules in future. You are having the structure. Why we have studied the structure of these molecules so intensively is this structure is very, very important in designing the drug molecules. See, we'll move on to the one aspect what we have studied is structure elucidation of carbohydrates. That is chemical methods, how elaborately they have done the structure elucidation of the carbohydrates. Okay. Years together they have worked and they were able to find out the structure of a simple glucose. Afterwards, they have established the structure of all the molecules. You are having the structure of simple sugar, then polysaccharide structure, you know the structure of amino acids, proteins, nucleic acids, fatty acids, sphingolipids, different varieties of things. Now we are having the structures, exact structure, their orientation, their three-dimensional existence, all these things we are aware of this. Then, after knowing all these things, where this can be used, the skill, where it can be adapted. Now we will move on to an aspect. This is called computer-aided drug designing. Computer-aided drug designing. What this computer-aided CAD implies, CADD. It is a process of drug discovery in which you are going to use computer, that is softwares are used to design the drug. Why we need new drugs? Because most of the drugs what we are using, they are of low efficacy. Some of them are not showing that much of effectiveness. They may have some side effects, and now we are having new diseases are coming, okay, and cost of production, okay, and industry has to survive, so they are also try to introduce new molecules into the market. So this is one aspect why we need new drugs. As I told, large number of diseases are there. Nowadays we are having large number of diseases that may be because of the habit, the food, what we are eating nowadays, okay, the quality of food, junk food we are using. We are addicted to so many other things which are not having healthy practices because of that, and environmental pollution, then lack of exercise, disconnection with the nature, okay, we are moved away from the nature, okay, and inadequate rest. All these things are resulting in new diseases, either physical, mental, whatever it may be. Okay, to overcome for these things, we need new drugs. We need new molecules which can act as drug. So where our computer aided drug designing is of very good use. Drug design is the inventive process of finding new medications based on the knowledge of biological targets. Computer-aided drug design uses computational approaches to discover, develop, and analyze the drugs. It is also identify biologically activity of the molecules. Okay, generally how the drug discovery takes place. Normal process of drug discovery, initially we randomly screen 5 to 10,000 to 20,000 of compounds are synthesized in the laboratory, and these compounds are randomly screened for different properties: antibacterial, antifungal, analgesic, anthelmintic, antipyretic, whatever the diseases are, for them all we are doing the test of these molecules. It involve target identification, validation, assay development, lead molecule generation. All these things is required, and the time period for this process will be approximately it will be 6 to 7 years. Afterwards, we'll be able to identify out of this 10,000 or 20,000 molecules, 250 molecules will be identified as lead molecules, and these lead molecules are further subjected to ADMET test, that is absorption, then distribution, metabolism, okay, excretion, etc. In vitro and in vivo test. All those things have been carried out for these molecules. Once this molecule passes this, out of 250, only 5 molecules will pass us. Again, this will take 2 to 3 years, right? As a drug candidate, and this drug candidate will be taken for further evaluation, their solubility, other parameters, its toxicity, etc. All those things are checked. Then preclinical phase. Here, in this stage, the molecules are subjected to different testings. This is phase 1, phase 2, phase 3. Then in phase 4 is actually, it is called as market biovisualization process. Okay, yeah, all those things will be done. Then it is subjected to market. The entire process will take more than 12 to 15 years. This will take 15 years to develop one molecule as a drug. And the amount involved, you just see, it is billions of dollars of rupees have been used to develop the drugs. Okay. Here, I'll show this one. This is the process. Disease has to be drug discovery process. In this modern discovery process, identify the disease. The disease is associated with a protein. You have to identify the protein. Now, once the protein is identified, then we have to look for a molecule which will bind to this. Very simple. If it binds effectively, then it can be used as a target for this molecule. Then it will go for preclinical trials, then FDA, etc. approval, and then it will become a drug. Okay. Here there are two variations we can see. Correct? So, as I told, this involves large amount, time, okay, all those things. With this note, I want to Okay, why CAD is very, very important. Drug discovery in this CAD will be done easily. Here we can save the cost, time, and uh, computation also will be less in this drug discovery process. This is a protein molecule. This is the drug molecule. Okay. See, we know paracetamol is a drug. Okay, it's an analgesic antipyretic. If you are able to link that one with a respective protein, which is responsible for elevation in body temperature or pain, okay, that will bind to this one. Protein binding will takes place. If the process of binding takes place, then we can take it as a drug molecule. Some other tests will be done, lead optimization, new drug can be discovered. This is the process. I will explain later. Okay, see, as I told, the CAD is very advantageous because it saves the time, it saves the cost, it increases accuracy. Okay, have more detailed information about the disease. Database screening is easy. No compounds have to be purchased or synthesized in the beginning. It is also possible to investigate compounds that have not been synthesized yet. Okay, all these things can be done with less manpower. Okay, using this drug discovery, these are the few molecules which have been identified, developed as a drug. Okay. Here, for this purpose, one has to know the structure of the protein or the structure of the drug molecule. The drug molecule, we will call it as ligand. If you know the structure of the drug molecule and protein to which it binds, okay, then it is called as structure-based drug designing. I know the structure of the protein. Okay, structure of the protein is unknown. Okay, I know the structure of the molecule which is useful as a medicine. Okay, that time this is ligand-based drug designing. So you are having the ligand, but you have to select the suitable protein where it binds. That has to be done using the computer and softwares. If you know the structure of the protein and the ligand structure is not known, then it is called de novo designing. If both are unknown, then it is this computer-aided drug designing is of no use. Then the traditional methodology has to be adopted. See, this is the drug molecule. Okay, here receptor, I don't know the receptor, so it is not binding to the receptor. It is not binding to the receptor. This is a drug molecule which is useful to the human being as a medicine. Okay, but I am unable to identify the receptor. Here we know the receptor as well as the drug. So the binding will be taking place, the interaction will be there. So this interaction shows that the molecule will bind to the protein properly. Okay, workflow of this CAD. Here in this workflow, okay, structure of ligands is known, various ligands, okay, of the structure is known. Let us assume that we know the structure of paracetamol. Okay, we know the structure of paracetamol, right? Simple structure is there. You can create varieties of paracetamol, chloro-paracetamol, bromo-paracetamol, like that. Different substitution can be done, and that can be used as a ligand. The protein is fixed. Okay, you can mix it and you can do the testings, etc., using the computer. If the binding takes place, then you can also calculate drug likeliness, what is its toxicity. Okay, then whether pan assaying interface compounds are there. This is pain is pan assaying interfacing compounds. Some compounds will mimic the things. They are not drugs, but they will show some values. So that can be and ADME, adsorption, distribution, metabolism, and excretion. How they are excreted from the body. All these things can be calculated using the computer. Once it works out, you have to rank it. The molecule has to be ranked. Okay, those molecules which are having good score, they can be taken as the drug molecule. They will be subjected to in vitro assay. So experimental on the animals and other things, they will do the experiment and they will finalize it and it will come out as a drug molecule. Okay, this is ligand based drug designing. See, as I told, a molecule, we know the molecule, right? This molecule, let us assume that this molecule is ibuprofen or something like that, which is a very good analgesic or antibiotic molecule. In this molecule, there is an aromatic ring system is there, aromatic system. These are hydrophobic groups are there. This is hydrophobic groups. Cationic group is there. Then hydrogen bonds are there. Hydrogen acceptor groups are there, right? This is an hydrogen donor is there. This pink color is hydrogen donor and this is hydrogen acceptor. These are the groups which are responsible for the activity of that ligand. Now, you know the drug, okay? Now, you know the structure of the molecules. So you have studied so many molecules, okay? Length and breadth, you have studied the molecules, which are biologically available molecules. You bring them, those molecules, aromatic molecule, you can introduce that aromatic molecule here, then uh, hydrogen accepting groups, donating groups, then anionic groups, that is negative end or cationic groups, okay? All those molecules which you have studied are synthesized, which are the structure is known, they can be brought and you can build a similar molecule of this type. Similar molecule can be built. So this ligand based drug design involves that type of skill. So to synthesize a similar molecule by knowing these things. See, this is commercially available molecules. These are all commercially available molecules. See, one molecule is there, the another molecule structure is also same. So aromatic ring is there, another aromatic ring molecule is there, but additional things are there. Some hydrogen bond groups, electron donors are there, electron repellents are there. All those things are there. You have to get a knowledge about the group's positions, where are the group the molecules are placed. Okay, so this group positions is very, very important. Okay, we may study the activity by altering the groups. One can, I told, so you can change the functionalities and you can study the activities also by replacing different functionalities. This is aromatic ring. Benzene ring is there. It may be hydrogen is there. You can introduce a chlorine atom here or bromine, okay, or OH group here. Then some repellent groups or adsorption groups, and you can synthesize different molecules. Okay, all these molecules can be tested against a protein to check whether that it binds to the protein. If it binds to the protein, it can act as a drug molecule. That's all. Very simple. See, even QSAR, that is quantitative structure activity relationship. This also can be studied. You can generate a formula for that. Different molecules we have to have a different formula. This is biological availability. This is log 1 by C is called as biological availability. How much molecule which is biologically available. All those things can be done by changing the structure of the molecule. So we can change the structure of the molecule. Okay. Now, identification of the protein is very, very important. For a particular disease, which protein I have to select? I am having the drug molecule, means I am having a paracetamol molecule, I am having whether it binds to the protein or not. That has to be done. So free softwares are available. If you if you type this one, http://rcsb.org. This is protein data bank. Okay, protein data bank. Here, this is a free site where you can go and download different proteins which are present in the cell wall, kidney tissues, heart, brain, where all our body is made up of protein. Different proteins are there. Okay, if one disease is there, bacterial infection is there, you can go to that bacterial cell wall, you take that in the cell wall, you or mitochondria, whatever, wherever is there, you can take out a protein. So proteins are available like this. Download this protein structure. Okay. So you will have the protein structure is will be there. Then to this protein, some receptors, all those things will be there. This is a ligand. This is the drug molecule. This is your protein. You put both of the structures into the software in the computer, and these softwares are also freely available. They will bind to one another. See, this is the binding area, how it binds and all those things. It gives you large information will be given. See, large amount of information will be given, how it binds, what is the efficacy, what is the amount of energy. So many data you are going to get. Okay, so based on this scoring, this is the lock and key model. Will say how it nicely binds. All those things will be. This is the scorings, right? These are the softwares which are available: Doc, Autodoc, Felix, Gold, Glide. This is Contingency software. Some of these softwares are free softwares, and this is a paid version of the software. You have to purchase the software to use it. Okay. See, this is the last one. Dinova, where we know the protein and we don't know the drug molecule. See, I told the protein, there is a structure. Okay, this is the concave place is there. Okay, this concave area, I know the area of this molecule. In this area, I will try to put one sugar unit, then I will put something else, then phospholipid I will put, some other unit I will put. Okay, like this, I will try to fill this area. Afterwards, filling these molecules with these molecules, I will link one another with some bridging groups. Some bridging group. I want to link these things. Okay, I may use CH2 or sulfur, whatever it may be. Okay, I will link this one and I will prepare a linked molecule of this type. So this linked molecule, okay, based on the protein site only, we have built this molecule. This will be your drug molecule which can bind to the protein surface. Okay, if it binds to the surface, now you are having the structure of the molecule. This structure, definitely it will bind to the protein surface and it acts as a drug molecule. Okay, I will take up one case study for this cancer. Right, in the treatment of cancer, prostate cancer treatment, this is a work is done by Professor Kabilan of Annamalai University. I have taken this one as a case study. See, these are the available drugs in the market. These are the available drugs in the market. This is flutamide, biglutamide, okay, azuletamide, etc. are there. So what they have done is, see, here there is an amide linkage is there. Amide linkage is there. There is an thiosulfonamide diazothiazole, okay, is there. That linkage is there. So here what they have done is they have simply created a molecule of this size. The size is very important for them. There is an aromatic ring is there, aromatic amide linkage is there. So they have taken aromatic amide linkage. Here CF3CN is there. Instead of this, they have taken very simple substituent taken and ring. And one more aromatic ring they have introduced. Okay, so this is the one of the molecule, simple molecule they have created. Okay, here complexity is there, NO2 group is there, CF3 is there, CF3CN cyano group, okay, amide linkage, so much things are there. But knowing this structural unit and their size, their shape, etc., they have written a molecule. An arbitrary molecule has been designed by them, and based on that arbitrary molecule we have told, no, so like this fluoro, bromo, etc., they have kept and they have synthesized some 10 to 12 molecules and they have been docked against a cancer protein. This is the binding. It nicely binds to the cancer protein which causes prostatitis, cancer, and acts as a drug molecule. See, these are the data which are available in the computer. Okay, see, they have synthesized so many molecules, out of which this is biglutamide is the drug molecule. See, the value for this is minus 11.01. Whatever the molecule they have seen computerally generated by applying their skills, their knowledge about the chemistry and other things. Okay, they have written the structure. That molecule is also having a very good value. So this is the binding sites are histamine 874 and tryptophan 741. It is binding. This is tryptophan 741. Arginine is 752. See, just a small modification will change the position of attachment. Okay, so whatever it may be, so this is binding to this proteins, and this is binding to the same 741 and 874 is there. Here it is 752, different amino acid units. These are the amino acid units. Okay, and it behaves as a drug, and its solubility is there, molecular weight is less than 500. These are the ADME absorption values should be there. This is a toxicity test they have done. Okay, toxicity test has given very good thing. And afterwards, what they have done, they have synthesized those molecules, whatever the molecule they have done, A to 1, A, B, C, D, F, G, H, I, J, K, L, M, N. Okay, or they can only synthesize the molecule 1G. Okay, 1G, which is having the good anti-cancer property. So the structure of that molecule, when it is subjected to in vitro studies, they have taken cancer cells. These are all cancer cells. They have added the drug molecule. The drug molecule is effective in killing the cancer cells. See, this is how the work has been carried out by knowing the structures of the molecules. That's why I say the structure of biological system, biological molecule is very, very important in designing the new drug molecules. This is what I wanted to talk today. Thank you very much.